Welcome, explorers, to the virtual conference chamber of our stellar biome starship. I am Kalam Bey. Today, we pry open the magic doors of biology's most enchanting factory, protein synthesis. Protein synthesis inside the cell. Are you ready? Then drop into your virtual belts, lower your voices to a cosmic register, and fire up our molecular engines. This podcast will feel like a sci-fi epic, yet deliver molecular biology tuition worthy of top-tier students. Don't forget code brainwaves for my Udemy course, SAT Biology 101, Unlocking Life's Blueprint, Level 1, packed with 3D printable models and high-res graphics to bring the mystery to your desktop. Subscribe and show your support for space mission biology. Now let's dive into the cell factor layers. Transcription, translation, and the engineering marvel of ribosomes. Like any factory, the cell starts with blueprints. The nucleus is the common center. Here, the glorious DNA blueprint is transcribed into RNA. We call this transcription. First of all, nuclear envelope and entry positions. DNA is safely locked inside the nucleus. The nuclear envelope acts like our ship's airlock, allowing large molecules through under strict control. RNA polymerase waits for the go signal to bridge to lock. RNA polymerases dance. We call it the molecular scribe. It unwinds the DNA helix and synthesizes an mRNA strand. Imagine a cosmic typewriter firing off 40 to 50 nucleotides per second. As your biology replicates billions of bases, this scribe sits across the genome at blistering pace. Promoter-based initiation. Every gene start line is a promoter region, a start command for RNA polymerase. Like an astronaut, go ahead to open the airlock. How tight the polymerase binds here dictates gene expression levels. Another section is, I'm talking about elongation phase. A polymerase moves, it rewinds DNA and releases mRNA. Think of processing a film reel seamlessly until the stop call arrives. This dance flows uninterrupted. Termination and capping. At the terminator signal, polymerase stops. The mRNA gains a 5 cap for ribosome recognition and a 3 poly a tail for stability. Now it's ready to exit the nucleus and journey to the cytoplasm. Thus emerges a fully processed mRNA, an interstellar message primed for the ribosome station. C now shifts to the cell's colossal assembly line, the ribosome. mRNA has arrived. tRNAs line up. Amino acid blocks snap into place to build proteins. Initiation complex. Ribosome splits into 40S and 60S subunits, assembling around mRNA and initiator tRNA carrying methionine. This is the initiation complex. Methionine is the universal start fuel, like priming a cosmic reactor. Codon-anticodon pairing. mRNA's 3-letter codons match tRNA anticodons. AUG, UUU, GGC. Each codon maps to an amino acid. Imagine 64 codons as galactic data packets directing traffic of biosignals. Peptide-bound formation, 23S. rRNA's peptidyl transferase activity links amino acids, releasing the previously amino acids. Think of a robotic arm fastening parts on a zero-gravity station. Cells from thousands of peptide bonds each second. Elongation cycle. A site, incoming amino acid, tRNA docks. P site, peptide chain grows. E site, spans tRNA exits. This 3-site choreography is a subatomic ballad, precision engineered. Termination, stop codons, UAA, UAG, UGA, summon release factors, disassembling the complex. The nascent polypeptide exits in its initial form. Folding and processing. Polypeptides fold into 3D shapes with chaperone proteins guiding them. Like industrial robots sculpting liquid metal, disulfide bonds and glycosylations deliver final functional touches. Thus, nanoscale factories forge and proteins fueling life machinery. Armed with these insights, what can we build? From CRISPR to biofuels, biotech is revolutionizing industry, machine, and space exploration. Recombinant protein production, insulin, vaccine antigens, industrial enzymes, all mass-produced in bacterial and yeast cell factories using these synthesis pathways. Synthetic cell factories lab-engineer entirely novel proteins, space-resilient enzymes, or microbes that convert waste into biofuel. These living machines transform raw materials into high-value products. future space colonies, portable bioreactors on Mars generating consumables, living modules at lunar bases supporting human crews. Soon our world habitats will thrive on engineered biology. Ethical and social dimensions. Editing human genomes or spawning new life forms demands responsibility. As scientists and planetary citizens, we must tackle these questions head on. Our journey through cell factories, the perfect mechanism of protein synthesis, comes to end, but for now. I hope you feel the gears, the robot arms, the wonder driving molecular assembly. Coupon code Brainwaves, SAT Biology 101 Unlocking Life's Blueprint Level 1 on Udemy, my course. Download your 3D printable cell model and host your own nanofactory at your desk. This channel is the cornerstone of my scientific documentary dream. Subscribe, leave your thoughts, and join me next for Epigenetic Beyond Our Genes. Let's drink coffee, friends.